Good evening. My name is Bella Lapp, and I am a student project manager at the Clark Forum for Contemporary Issues at Dickinson College. Before we begin, I would like to take a moment to acknowledge that Dickinson College is on the unceded lands of the Susquehannock Nation. We hereby recognize the prior status and enduring diversity of this land and the many indigenous nations that stand in relation to it, particularly the thousands of indigenous children from dozens of tribes forced into the reprogramming camp established at the Carlisle Indian Industrial School starting in 1879. Dickinson College supported this agenda of cultural eradication in both word and deed. Turning honestly towards that shameful past animates this acknowledgement and gives orientation to our desire for a reconciled future. Accordingly, this living land acknowledgement is intentionally incomplete, a reflection of the ongoing process it represents, to learn respectfully from the stories of this land and the peoples that carry them, to think reflectively about the injustice in our shared past, and to act responsibly with that knowledge today to inspire a more equitable tomorrow. On behalf of the Clark Forum, Dickinson College, the Wellness Center, the College Farm, the Food Studies Program, and the Departments of Biology and Psychology, I would like to welcome you to tonight's event, exploring the strange and ancient biology of the brain hidden in our guts. I would like to highlight that tonight's program was student-initiated. At the beginning of every semester, the Clark Forum student employees propose ideas for programs focused on contemporary issues. We then vote to select 2 programs to develop for the following semester. Last fall, one of our student project managers, Natalia Fedorchak, proposed an event on the gut-brain axis. To pitch the event, she immediately grabbed our attention by sharing that we all consume about a credit card's worth of microplastics a week. Personally, that was all I needed to hear. She had my vote. Natalia graduated early, which we are all very proud of her for, and was not able to oversee the planning and preparation of tonight's event. As someone who never really leaves the humanities side of campus, I gladly volunteered to take over, excited to dip a toe in neuroscience. I am very grateful to Natalia for consistently advocating for more STEM programming. Tonight's event exemplifies the unique mission of the Clark Forum, which is to bring a truly interdisciplinary liberal arts perspective to contemporary issues. Developing this event has encouraged me to explore topics that I would have otherwise never been exposed to, and I hope it will do the same for other students and community members. The emerging research on the gut-brain axis is increasingly relevant to our generation, as we are forced to confront the long-term effects of plastic waste and food sustainability. Subhash Kulkarni is a PhD scientist who currently serves as an assistant professor of medicine at the Beth Israel Deaconess Medical Center at Harvard Medical School. He is also a member of the Neurobiology graduate program at Harvard University. His National Institute of Health-funded lab at Harvard University, where he serves as principal investigator, studies the complex biology of the enteric nervous system and the gut-brain axis. He has completed 2 postdoctoral fellowships, one in structural biology at Stanford University and one in enteric neurobiology, also at Stanford. There will be a question and answer session immediately following this program, so please hold all questions until that time. The Clark Forum welcomes differences of opinion, expressed politely, thoughtfully, and succinctly. Disruptive behavior or harassment of the speaker, members of the Dickinson community, or audience members will not be tolerated. As a show of respect for our speaker and everyone in attendance, please stay until the end of the program, including the question and answer session. In the event of an emergency, please note that accessible exits are located on the west side of the building, back there. At this time, I ask that you silence all cell phones and electronic devices. And now, please welcome Dr. Subhash Kulkarni.
Thank you so much. This is such a great pleasure to be here. I had an extremely enjoyable day talking to everyone in the student community as well as in the staff. And it's very touching to actually come and meet all the undergrads, and especially undergrad education, see flourish this well and this beautifully at Dickinson, which is really a foundry for the next generation of scientists and freethinkers and artists and literary figures. So I'm really touched for this graciousness, for inviting me to Dickinson. And here we go. So the title of my talk is Exploring the Strange and Ancient Biology of the Brain Hidden in Our Guts. Now, I purposefully kept the talk to be slightly intriguing, slightly strange, slightly mysterious, because I really wanted to make it not appear pedantic, but start and cater to an audience which is a diverse audience, as you are at Dickinson. Before we get started, I just wanted to note that one of your alumni says hi. She's in my lab. She's a research tech and lab manager. It's Violet Hong. She just presented work at her first conference. So we're happy to report that this is the second alumnus that I know of now from Dickinson, and that she's doing well. So here we get started. So after I got the invite from Dickinson, I was reminded of my time as an undergrad. And one of the first questions that we asked ourselves is, what is life? We did not come up with a satisfactory answer. It's a question that has perplexed the most brilliant minds. But we thought we made some headways into it. And when we started thinking about what is life and how do you define life, the easiest way that we came across with is, let's consider life as a system, which is a closed system, in a very biophysical characteristic. It's a closed system in which the entropy of the system decreases inside the system at the expense of increasing it outside of the system. Now you can think about reproduction or anything, and then I can point out there is also crystals that propagate. So a crystal is not a living state. But the most succinct definition that we could come up with when I was an undergrad is a closed loop system in which the entropy decreases inside the system at the expense of increase in entropy outside of the system. And how does a cell do that, or how does a living organism do that? It does it by metabolism. So it is able to create complex biomolecules using very simple molecules, using sugars, and create large structures like DNA and protein. So the act of metabolizing or anabolizing, creating these larger biomolecules, is an act of reducing entropy. And the way that it does it is by using all the energy outside of the system. So metabolism is key to life. You cannot have life without metabolism. And what is key to metabolism is digestion. You cannot have life without metabolism. You cannot have metabolism without digestion. And let's think about what exactly I mean by all these things. So this is an interesting system where we took single cells from an adult mouse gut. This was just one particular small layer. And we just started saying, well, what exactly happens if we start growing it up together? And you can actually see that in this video, you'll see that there was some kind of a self-organization that these cells underwent. And there was a lot of things that the cell started doing, things like movement on its own, autonomous movements, autologous movements. But they could not do it unless I started providing a glucose source. So there was digestion, there was metabolization of these complex things that was happening, and that really catalyzed all these cells to start coming together and start observing, start showing, start exhibiting some kind of a complex behavioral trait. Now, you can actually take the simplest of the systems without sugars, without anything that the cells can digest out. These things can't happen, which means that there has to be an ingress of energy for increasing or decreasing the entropy inside the system, and there has to be expulsion of materials that are waste products, which takes us to this beautiful thought that at least in the vertebrate world, or at least in the living world, everything eats and everything poops. And you can start thinking about and come up with rational ways, and it's like, oh, hang on, plants don't exactly poop, do they? But they do, because they are producing a lot of waste materials that are expelled out. Carbon dioxide is a waste material. So when I say poop, it's not the poop that we know of intricately so, but in all sorts of waste materials that are expelled out. So the crux of life after having a beautiful dinner outside, we can attest to that. Everything eats, everything poops. So let's look at this across the evolution of metazoa from single-cell organisms towards more and more complex organisms. So you can actually see a choanoflagellate here, and then it goes into sponges. You can actually see that there is an ingress of materials coming inside, and these cells are having access to these materials, and they digest them out. They take all the nutrients that they have, and then everything that is a waste product is then it's expelled through this egress. And then you can do the same. You can look at the same thing through ctenophores, where there is a particular mouth and there is an anal pore. So things are taken in and things are expelled out. And these are very simple systems, but the crux of the matter remains. Life depends upon your ability to utilize nutrients and expel waste products, your ability to eat and your ability to poop. Any of those things that is impacted negatively is a life-ending game. And you can actually see this in the placozoa as well. There is algal food that these start ingesting now through specialized lipophilic cells and gland cells. And then you can go on to much more complex organisms. You can have the insect, where there's a foregut, which is endodermal derived, and there's a midgut and a hindgut. They do different sorts of things. And then in mammals, you have, again, the glycocalyx and the enterocytes. So the systems can become more complex over evolutionary time, but the crux remains, is you have to have an ability of ingesting food. The complexity of the food, the complexity of the ways in which we go and hunt for food, the complexity in the way in which we digest food, that all changes. The simplest of the simple things remain: eat and poop. So if you think about complex systems or even simple systems, every function requires regulation. And that can happen at the level of single cell as well. If you have a single cell system where you actually are digesting food, which is sugars, then you need a regulation of that particular biochemical pathway that allows you to digest that food and take in the energy. You can take it from a single cell level to a complex level. Every function requires regulation. And as we become more and more complex, as life became more complex, as there was more and more division of labor, these complexities of function, the regulatory functions, they started grouping together in groups of cells that we started annotating as neurons. So neurons are basically cells in a metazoan body that are performing regulation of functions. And these are various regulatory mechanisms. They can be regulation of thought in higher order animals. They can be regulation of digestion, as in the enteric nervous system. They can be regulation of interoception, nociception, which is pain signaling and pain sensing. But regulation is fundamentally provided by neurons. So as we start thinking about now neurons as the major force, that is regulatory force, let's consider this one beautiful paper, a review article that was published by our colleagues in Melbourne, that the first brain was the brain that was geared towards digestion, because the most fundamental life activity is digestion. Everything eats, everything poops, which means that particular function, that is the most life-giving function, the most central to life, requires a particular neuronal organization and neuronal regulation. And you can actually see this in all the animals. You can go to the hydra, you can go to echinoderms, you can go to octopuses, you can go to humans. Every single system has a neuron or neuronal-like cell body that is performing intricate regulation of digestive functions and gustatory functions. Now, if you consider we all know of neurons, and everybody, if you close your eyes and you say the word neuron, the first thing that comes to your mind is the brain. Now, if you consider an alternative explanation, that in the lower order animals, in the most simple systems, or in sponges, for example, there was no locomotion. There was hardly any executive function. So if you think about it from a most reductionist model, that this is what the nervous system in lower order animals looked like. There was a very, very little motor function, if any. There was very little executive function. Most of it was geared towards digestion. And as we go through evolutionary time, you can actually see that organ systems became a little bit more complex, allowed the evolution of executive function and locomotory function. So the amount of executive and motor function started increasing in these nervous systems. So much so that things started diverging. There were two different nervous systems now that were formed from one nervous system. There was one that was going more and more towards locomotory functions and the executive functions, and the other remained hardcore digestive function. So much that these two became completely independent of each other. And as they completely became independent of each other, they started being located in two totally different parts of the body, where the digestive function remained in the enteric nervous system. There was a new nervous system that evolved, which was completely dealing only with executive and motor function. That is now what we call as the central nervous system. And then there was a connector that's between them, because they were always connected. Just the fact that they moved away, the neural networks that were still feeding both of them or still making the crosstalk between these two nervous systems still remained. So this is what we call as the gut-brain axis. So you can actually see the case that I'm making over here is that while everything else changed in the central nervous system, things changed in the gut-brain axis. The enteric nervous system is and remains the most ancient and the primordial nervous system, because it is catering to some of the most basic aspect of life, the most basic aspect of life, which is digestion and metabolism. Everything eats, everything poops. So if I show you this particular diagram, you will say, nah, this sounds far-fetched. But let me show you what exactly the central nervous system and the enteric nervous system and the gut-brain axis looks like. This is exactly what it looks like. The enteric nervous system is all in the body of the gut. The central nervous system is far removed from that in the brain, brainstem, and the spinal cord. And there are all these pathways, all these neural networks that are connecting these two. The CNS, the executive and motor function, the digestive function in the enteric nervous system, and the gut-brain axis. That includes the vagal pathways, that includes the sympathetic pathways, and that includes the pelvic pathways. So this is the evolution of the nervous system that we now know as. We are very cognizant of our central nervous system because it is catering to a cognitive ability, it is catering to a motor ability. But we are not that cognizant of the most autonomic pathways, and the largest subdivision of the autonomic nervous system, or the peripheral nervous system as we know it, is the one that remains inside of the gut, which is called the enteric nervous system. So the brain that is hidden in our guts is the enteric nervous system. So what does it look like? Now, this is not an animation. This is not ChatGPT generated. This is actually a real 1 centimeter piece from a mouse intestine where all the neurons have been labeled in red. This is what the enteric nervous system looks like. And if you actually look at it, it's pervasive. It's actually going in all different layers of the gut. In the mucosa, as we are going through it now, and it forms these concentric circles and these concentric networks. So it regulates almost all functions of the gut, including motility, digestion, absorption, secretion, immunity, hormonal regulation, and even metabolic and systemic regulation. There are about 500 million neurons and about 2 billion glial cells in this nervous system. They all are located inside the gut. There are concentric networks. There are two different networks. The largest one is called the myenteric plexus, which is the one that you see over here. You can actually see the chicken mesh wire-like the structure that emerges. And dysfunctions in this nervous system are associated with or cause the various and diverse gut dysfunctions that we know of. This is what it looks like on a scanning electron microscope. And every single clump that you see over here is called as ganglia, and then there are strands that connect one ganglia to the other ganglia. And this is my image where actually you can take one ganglia and look at it under the microscope, and that's what it looks like. So you can actually see there are clumps of cells sitting in here, and these are the neurons and the glial cells, and they're connecting through these networks to the adjacent ganglia, and then to the adjacent ganglia, and then so on and so forth. There's about 21 centimeters of the intestinal tissue in mice, and every single piece looks exactly like that. So there is a structure here that we just don't understand. We can understand the structure of the cortex, of the hippocampus, of thalamus, hypothalamus, subventricular zone. We know these different regions and what exactly the functions that they do. We have absolutely no idea about how these are different over repeated segments of the gut. But this is the ultrastructure of the enteric nervous system to give you a glimpse. So now imagine that there is a nervous system that is that huge, that is that significant, and it is located in an organ that is constantly moving. The strain and the stretch on this nervous system is so immense that if it was incumbent on your CNS, you will actually get a concussion. It is millimeters away from toxins, microbiota, all sorts of viruses, all sorts of drugs that we take, medicines, all the sorts of McDonald's food that you eat, every gulp of Coke and Diet Coke and Pepsi, and yet it works unfailingly over years and years. It works unfailingly from the time that we're born to the time that we die, mostly. So it deals with a lot, but how is it maintained? So there's mechanical, biotic, and abiotic insult. And as compared to the CNS, where there is a skull, that is, there is a protective layer, there is a blood-brain barrier, there is a meninges that really protect the brain, there is really nothing protecting the gut, the enteric nervous system in the gut. So then the biggest question that we started off, this is way back in 2008, is despite all of that, how is the adult enteric nervous system maintained? And I came up with two different plausible explanations for this. Is that either there is a strong protective force that exists, that I don't see, but maybe there is, that really allows the exact same neurons to be maintained. The same neurons that you're born with are the same neurons that you die with. That it's really protected somehow, but I don't know exactly how. Or everything everywhere is new. It's a constant way in which this is being replenished. Those are the only two explanations I could start with. So it took us a few years to figure out how to exactly answer this question, what kind of experiments to do to answer these questions. And when we started doing that, and I'm going to take you through a very simple way of showing you some of the data. When we looked at it, we actually saw that by using immunohistochemistry, where you take antibodies against particular cell markers, you could actually see that if you label all the neurons, shown here in green, with a particular antibody, and you can detect it, and you can visualize it, and you can visualize their nuclei, shown here in dye, with the nuclear labeling dye. And along with it, you take an antibody that is directed against a marker, a protein that is only expressed by dying cells. It's called apoptotic cells. Then you can actually see that you can very easily detect that there is a significant proportion of neurons, very easily detectable, that is dying all the time, because they're expressing this apoptotic marker that we call as cleaved caspase 3. And cleaved caspase 3 is expressed by all these neurons, as you can see here in the yellow arrow, while there are other neurons shown here in the white arrow that don't express this. So there is a subset of neurons that is dying, and there is a subset of neurons that is not dying. So let's then figure out how many neurons are dying all the time and how many neurons are not dying. And when we looked at that, it was astonishing that we found that at any given point of time, about 10% of neurons were dying at any given point of time. Now 10% is a huge number, because if 10% rate goes unchecked, in 10 days, you have no more neurons, or less than that, because it's a regression. So at any given point of time, 10% of neurons are fated to die. And yet, the ENS remains stable, and it carries on years and years, days upon days, no matter what. So how is this maintained? So something lost has to be made again. Le neuron est mort, vive le neuron. So for a neuron that dies has to be reborn again. So how does it happen? So if you think about how new neurons can be generated, it happens through a process called neurogenesis. And since this is happening in adults, not in the fetal tissue, it is called adult neurogenesis. And the simplest way of explaining that is that you have a neuron, and there is a cell that is generating this neuron. And since it's a precursor cell that's generating this neuron, it's called as a neural stem cell. It's a stem cell that proliferates, that keeps itself alive, but occasionally it gives rise to a neuron. It can't be occasionally, because we are seeing a rate of neuronal death that is 10%. So it has to be a fairly high rate of neurogenesis. So we started asking the question of what exactly is this neural stem cell? Is this really present in the gut? So we took a few years and figured out that the whole community knew there is one particular protein called nestin, which is expressed by neural stem cells. So we started asking the question, is nestin present in the enteric nervous system? And when we looked at it, we actually found that within the ganglia itself, right next to the neurons, there are these nestin-positive cells seen here in green. I'm assuming that this is green, but it's labeled with the green arrow. And then the neurons are here in magenta, and the vasculature is labeled in red. So there is a neural stem cell, or a cell that expresses a neural stem cell marker, that's sitting right next to a neuron. And we're like, oh, that's interesting. So if you have the neural stem cell right next to a neuron, can that be that this is regenerating neurons in adults? And long story short, when we actually so there is an indelible way of marking cells, and that's called a Carlisle system. In simple terms, it's basically you label every single cell that is expressing a particular marker with an indelible dye or indelible fluorescent reporter, and you can detect that. So the cell may stop expressing that protein, but it will be forever labeled with that dye. So when we labeled all the nestin expressing cells with red, here you can see the red arrow on the left hand side, and we labeled all the neurons in green, and we let the animals be for some time in another cohort. In some time, we actually started seeing that the red positive cells, nestin positive cells, remained as nestin positives, but they also gave rise to other cells that were expressing both neuronal marker as well as nestin. So they were both red positive and green positive. This basically means that this green positive neuron, this green colored neuron, was coming from a cell that was nestin positive just a few days ago. And when we quantified the numbers, over time, we actually saw that there was an increase in the number of neurons, in the proportion of neurons that was coming from nestin positive cells, suggesting that here we have evidence that nestin positive cells are giving rise to neurons. So new neurons are being generated from stem cells that are expressing nestin. Not just that, but even the rest of the cells, the nestin positive cells, their numbers are increasing. So here you have a perfect explanation for a stem cell. It not only generates the cell it's supposed to generate, but it keeps on proliferating to generate its own numbers up. So here you're seeing a characteristics of a stem cell being met, where it's not only generating the neuron, but it's also maintaining its own numbers. So we then followed it up with other experiments where we labeled the DNA with chlorinated deoxyuridine derivatives. This is a method of seeing over a course of time when you're giving this particular drug, how many cells are being generated. It's a very simple assay that you just trick the DNA into accepting one of the donors, one of the drugs, so that every time the DNA replicates, you have this particular drug that is accepted into the DNA. And the beauty is that there are antibodies through which you can detect this particular drug. So we used two different drugs. One was IDU and CLDU, and these were given at two different time points. And when we sacrificed the animals, we actually saw that there was IDU and CLDU in neurons that were in the mesenteric ganglion. Now, the way to understand this is these neurons, the ones in blue, that show both green and red, show that in the days that we gave this particular drug to the animal, the DNA was labeled. So the DNA was newly synthesized. So these neurons in blue that also show red and green nuclei are the ones whose DNA has replicated in the last 13, 14 days. And when we calculated how many of these neurons over 12-day period, 14-day period showed evidence of DNA replication, so suggesting that they were newborn, we found that about 88% neurons in a 2-week period were newborn, which when we did a math on that, it came out to be exactly 10% of neurons. So remember, we had seen that 10% of neurons die every day, and we see evidence that 10% of neurons are born every day. And this keeps the number of neurons in the entire adult enteric nervous system constant. So you have the rate of death counteracted by the rate of birth. So you have a constant replenishment of neurons to replenish those neurons that were dying out. And this keeps the enteric nervous system healthy and supple. So to summarize the first part, the adult ENS is constantly in a flux. It is constantly being replenished by two continual and co-equal processes, the one of loss and the other of genesis. It's almost like Star Wars, right? I mean, you have the force that is constantly being maintained. Then the constant supply of new neurons keeps the ENS young and supple and disease-free. So if there is any defective neuron, it's not going to last much because it's not going to cause much of a disease, because in about 12 days, it's just going to be taken out of the circuitry. So the defects of the ENS at that point of time are no more defects of neurons, but they are deficits or issues with the neural stem cells. So I'm going to segue away from here because this is a story that we published in 2017. It's very dear to my heart. But this is something that is a story that we just very recently published. And if you thought that as compared to the central nervous system, where people don't see significant adult neurogenesis, that the ENS is strange, hang on to your seats, it's going to get much weirder. So 2 movies came out in the last 10 years, which have been very close to my heart. The first was this, Inside Out. There's a second one coming up, which is that we spent a lot of time thinking about how the upper brain matures. We made movies about it. Pixar made millions of dollars from it. But there is absolutely nothing about that helps us understand how the brain in the gut matures, how the enteric nervous system matures. Now, we've all been kids. We've all realized that our feeding habits have changed during the time when we matured. We went from a very simple milk-based diet to now eating stuff that we eat as adults. So something changed, and somehow our nervous system in the gut geared and matured to accept all those food, accept all those stuff, accept all the drugs and chemicals and cokes and diets and stuff that we now do. And yet, somehow, we are absolutely fine. We understand how cognitive changes happen. We see a baby as the baby matures, and we say, oh, now he started speaking or she started walking. Those are things that we can readily understand, but there is really nothing that we know about how the enteric nervous system matures. Now, this is the second movie that I really like. We know not only not know anything about maturation, we know nothing about senility of the gut. We can see senility in people by their cognitive and locomotory changes, but we do know that humans become senile in their guts. 85% of patient visits from elderly in the hospice system are because of constipation, and we have nothing. People come to us asking, why is this happening? And we had no clue for that. We had no answers for that. So I decided that being a marked man in the 2017 paper was not enough for me. So I decided to foray into asking these two questions. And this was kind of timely because this was around the same time when my son was born, and I was seeing all these changes happen in his gut phenotype. And I asked the question, why? So how does the ENS mature, and how does the ENS age? Now, to take you back a little bit into developmental biology, there are fixed codes for where every single cell in the body comes from. Muscles are supposed to be coming from the mesoderm. Neurons are supposed to come from the neuroectoderm. The skin is supposed to come from the surface ectoderm. The blood is supposed to come from the mesoderm. The vasculature is supposed to come from the endoderm. So there are fixed codes that developmental biologists spent years upon years, close to about 80 years, working out exactly where every single cell in your body comes from. So that's why every time we talk about things, it's a mesodermal derivative or it's an ectodermal derivative. And the developmental biologists would, if you just give them an opening, they'll spend the next 6 hours talking to you about that, which I'm not going to do. So during fetal development, the neurocrest is the sole developmental lineage for all enteric nervous system cells and glial cells. And to give you an example, this is how it happens. So the migratory neurocrest is here. It migrates down when the embryo is very, very tiny. And it goes through this whole cascade where it gives rise to a neural precursor cell and a glial precursor cell. The neural precursor cell gives rise to a neuron, and the glial precursor cell gives rise to a glial cell. And so we thought, this really happens during fetal development. So this set the dogma that the neurocrest continues to be the only developmental lineage for all ENS cells, not only during the fetal phase, but also in all postnatal phases. Now this is the chart that I was talking about. And if I take a moment, you have the primitive endoderm that gives rise to all these cells, and these might look like extreme Greek and Latin to you, which it certainly did to me when I was starting off. But there is an algorithm, there is a chart that developmental biologists made up for many, many years, saying exactly where every single cell in your body, every single tissue is going to come from. And the neurocrest, as you can see, it's part of the neuroectoderm. Neuroectoderm gives rise to two different types of cells, so the CNS, the entire CNS, and then the neurocrest gives rise to the entire peripheral nervous system, the enteric nervous system, the cartilage and the bone and the melanocytes. The skin cells come from the same cells that gives rise to the peripheral nervous system. So as a corollary, not only did we know, but did we think that this was the dogma for the development of postnatal phase, how the ENS matures was thought to be a very straightforward extension of fetal ENS development. So the dogma was that development just doesn't stop during when the child is born or the animal is born. It's the same program that continues over the course of its postnatal development. So maturation is just a simple extension of the fetal development. It's just unfinished business that is still going on. However, this dogma was never tested. And hence our understanding of how the ENS matures and what are the molecular processes that separate from fetal development were very limited. So because of this, we had simply no understanding. We cannot come with a simple answer to the question, well, why is it that certain diseases happen only after the child is born? Why is it that certain diseases happen only before the child is born? So why are there certain congenital issues that don't come up during development, fetal development, and they only manifest during the postnatal phase of life? We had no clue about that. And we currently had no understanding for how the ENS ages. So these are the two questions that we tackled, and the exuberance of youth, or the stupidity of youth, as you may call it, made me test this dogma, and we started on this project somewhere in 2011. And it's a tale of 13 years of scientific pursuit, and I'm happy to present this to you. So again, just to clarify, the vertebrate neurocrest development happens as the neural plate border imaginates. This is the pre-migratory neurocrest, and it starts migrating out to give rise to the entire enteric nervous system. So we used a mouse model that allowed us to label every single cell of the neurocrest, and we called this the Halloween mouse model. And it's called the Halloween mouse model because it actually looks like a Halloween mouse model. It's where we are expressing a fluorescent protein called TT tomato under all the cells that are Wnt1 expressing cells. Wnt1 is a marker for the neurocrest, so it indelibly labels every single neurocrest derived cell. And we call it the Halloween mouse because when you shine a black light on it, it actually glows like this, because all the melanocytes are coming from the neurocrest. Since all the melanocytes are coming from the neurocrest, they all glow red when it's under a black light. So when we tested this mouse, we found that in the myenteric ganglia, there are two different types of cells. So remember, there's a neuron cell and there is a glial cell. So all the glial cells continued to be derived from the neurocrest, because all the glial cells shown here in green are also red, as you see in the red arrows. So all the glial cells are derived from the neurocrest. So that completely matches what our expectation during fetal development is. However, if you look at neurons here, you can actually see that neurons here labeled in the green immunostain, not all of them are red. So there is a proportion of neurons, green cells, that are red, and there is a large proportion of neurons that is not red, suggesting that they are not coming from the neurocrest. Now, we do know that at birth, everything is coming from the neurocrest, but something happens in between then and adulthood that really changes this paradigm. This is not supposed to happen, but it is happening. And when we quantified the numbers, we found that it's not a small number. 50% of neurons in the adult enteric nervous system are coming from a place that is not the neurocrest. This is not supposed to happen. Now, remember the chart where I spent some time talking about where the neurocrest comes from, and it comes from the neuroectoderm, and that all the enteric nervous system is coming from this? Now, so we see that this is not happening. And then you have the surface ectoderm, that is given the epidermis or the skin, that you have the definitive endoderm, mesoderm, that is muscles and bones and connective tissues. So I spent about 6 years trying to figure out all the different combinations in which I could explain this, and I could not come up with anything. So somebody asked me a question today about negative data. The answer to when you find negative data is just to keep going, because the negative data is not negative data. It is a pathway towards something, and I'll show you why. So on a Friday evening, when you're frustrated, what's the next thing that you do? You either go and drink a beer or you do something very stupid. And that's when I did something very stupid, which was I went to a next door lab, which was a cardiomyocyte lab, a mesoderm lab. And I said, can you just give me one of your mesoderm Cree lines? This is a line in which every mesoderm cell that's giving rise to cardiomyocytes and muscles and so on and so forth is labeling every single cell that is coming from the mesoderm in green. And when I took this mouse and I labeled the myenteric neurons of this mouse line against neuronal marker, I started finding that, lo and behold, not only is the muscle coming from the mesoderm, which is expected, but there is a population of neurons here in red that are also coming from the mesoderm. This is not supposed to happen. This was on a Friday evening. I could not sleep the whole weekend. And then we started looking at this, whether this is something that we can reproduce in other mouse lines. So we took about 5 or 6 mouse lines, and we're seeing exactly the same thing happening over and over again. And then we started asking, well, can we juxtapose the neurocrest derivatives and the mesoderm derivatives using an immunostaining system? And this was the killer experiment, where we took the neurocrest line, everything labeled in red is neurocrest, everything labeled in green is mesoderm. And you can actually see that there is this beautiful two neurons sitting in here that are cross-talking to each other. There's a connection in between both of them, shown here in the white arrows. They are coming from the mesoderm, and they're not coming from the neural crest because they are not red. So this was the killer experiment that really told us that, yes, this is really happening. And then we quantified it. We actually found that 50%, we remember we had found 50% of neurons were not coming from the neural crest. In this mouse line, we found that 50% of neurons are coming from the mesoderm. So the enteric nervous system in the adults is made up of 50% of neurons coming from the crest, where they're supposed to come from, and 50% from the mesoderm, which gives rise to muscles and cardiomyocytes, where they're not supposed to come from. So this is all that we knew about mesodermal neurons, and since we are in tax season, I thought I would just keep it over here. People who get their W-2s would remember this. But we know nothing about mesodermal neurons because this is not supposed to happen. There is absolutely nothing in the literature of saying this is what a mesodermal neuron is supposed to look like, except in C. elegans, interestingly, where there is only one neuron that is going to the gut, and that neuron is mesoderm derived. Which is very interesting. It's very evolutionarily conserved. So we started doing a lot of molecular characterization of the neurocrest-derived enteric neurons, which we started calling as NENs for the lack of a better phrase, and mesoderm-derived enteric neurons, or MENs. And we started doing all these immunochemistries, and we started finding that there was some significant differences, that the neurocrest-derived enteric neurons have more nitronergic neurons and cholinergic neurons, and the mesoderm-derived neurons are more sensory in nature, and it really required us to do in-depth classification and characterization, which we did. And we came across this putative scheme right now, which really shows that these two nervous systems that are co-locating and coexisting in the same space have totally different functions. The neurocrest neurons are sensory a little bit and motility regulating. They regulate the motility of the gut. And the mesoderm neurons are doing way more than that. They're regulating the extracellular matrix, how much collagen is supposed to be in the gut. They're releasing hormones, they're releasing cytokines, and all sorts of things. They're interacting with immune cells in ways that we did not think it was possible. So we started looking at this, and we started saying that, well, can we find markers for this? So for example, the sensory markers, we could find they were expressing CGRP, and then they were releasing ATP as a way of, they're releasing ATP as a neurotransmitter. They were releasing all these cytokines, as I mentioned, ECMs and hormones. The growth hormones were being released by the enteric nervous system, mesodermal neurons. So we were left at that point of time with this perplexing question. Well, they're not supposed to be, they're not coming from the mesoderm at birth, and in adults they're 50%. So something's happening between when the animal is born and when the animal is adult. So what does that timeline look like? When exactly are they coming into the gut? When are their proportions changing? How does and then after maturation, is it connected to aging? So when we did this experiment, we took mouse lines that were at different ages, and we quantified the proportions of the neurocrest lineage neurons and the mesodermal neurons. And this was very interesting. So just around P11, when the mouse is just opening its mouth up to stuff that is other than milk, and it's just started nibbling on solid food, we start seeing the first emergence of the mesodermal lineage. Their proportions then go to P22, and I'll actually just make it easier. At infancy, it's about 5% of neurons, very, very small. As the mouse becomes juvenile, this proportion increases from 5% to about 30%. And this is just in a 10-day space, 10 or 11-day space. Their numbers rapidly expand, and this is maturation. In adulthood, no matter what age I look at in the adulthood, their numbers remain constant, 50%. Except when the animal starts aging. So in a 17-month-old mouse, there is no more neurocrest neurons left. So the senility of the gut in the aging phenotype and the juvenile phenotype of the gut are exactly the opposite. The juvenile mouse is predominantly the canonical lineage, and there is almost a complete loss of that lineage in the aging mouse. In the maturation phase, this increase of the mesodermal lineage, this is a 6-fold increase in a space of 10-11 days. This has to be the craziest cell expansion in a healthy tissue that I have ever seen. This continues during between the juvenile phase and the adult phase. Is still about a fold-and-a-half increase, and then this obviously this is a crazy thing that I still am trying to reconcile with. So what exactly happens during this phase? So if I go into a little bit into the mechanics of this, one of the most crucial growth factors for the neural crest lineage is this growth factor called glial-derived neurotrophic factor. It's something that happens to be also associated with changes in Parkinson's disease. In Parkinson's patient, there's a reduction in GDNF level. So GDNF is one of those proteins, one of those growth factors whose levels are extremely important for the sustenance of the canonical kind of neurons. And if you look at what happens to GDNF levels in the same tissue over age, between P10, which is the animal this age, about P11, the infant animal, and the juvenile and adult phases, there is a complete loss in the presence of this protein. So we ask the question if this is an association or this is a causation. Because if it is a causation, then if you reintroduce GDNF back in, you should be able to arrest the development, arrest the maturation. That's exactly the experiment that we did then. We said, well, what happens when we give GDNF to the mice? The total number of neurons remains exactly constant, but look over here. The control mice show an expected phenotype of the proportion of men's, about 30%, 25 to 30%. But when they give GDNF, their numbers are arrested to the levels of when they are infant. So if you are adding GDNF back, you can actually arrest the maturation or development of the mouse. So here you have now a putative drug through which you can modulate the two different lineages. You can control what exactly is happening between these two nervous systems. The big question was, if this is true for during maturation, can you do this during aging? Because in the aging mice, we know that the aging mice have significant delay in their transit times. They have a constipation phenotype. So when we did that experiment, these are after we gave GDNF, there was a reduction in the proportion of the mesodermal neurons. But not just that. Look at what happens to motility. The motility is significantly delayed in both cohorts before we gave the GDNF treatment. And after we started giving the GDNF treatment, the mice had an almost completely normal motility. This is the first putative therapy that we have that is based upon a mechanistic understanding of biology that aims to relieve and cure constipation in the elderly. Just because, and it all started with a fishing expedition. So this is our model. Our model is if you have just right GDNF levels, you are healthy despite having a few scruffs. You're absolutely normal, you're ready to fight. That's what the enteric nervous system looks like when you have absolutely normal levels of GDNF. If you have too much, you have arrested development, you don't have maturation as it's supposed to mature. If it's too little GDNF, then you have this condition of accelerated aging. You actually age much more than your chronological age is supposed to. It's almost akin to writing an NIH grant these days. So to summarize the second part of the talk, ENS maturation is a process that is developmentally distinct from the fetal development of this organ. This is driven by origin and expansion of a newly discovered lineage of mesoderm-derived enteric neurons. The lineage ratios of the two lineages are indicative of age and health, and they have significantly different functions that they cater to. But there are some functions that are conserved. And aging is an extension of maturation, but depending upon the drug, you can regulate it. So, but a caveat over here to remember, aging is not lifespan. You can change aging to change health span, but that does not mean that it will translate into lifespan. So with that, I would like to take the occasion to thank members of my lab and all my collaborators all over the US and the rest of the world. With my funding acknowledgements, I'm happy to answer any questions.
It is now time for the question and answer session. So that everyone in the audience and our speaker can hear your question clearly, please raise your hand and wait for a mic to reach you before asking your question. We reserve the first questions for students, and then we will open up to the rest of the audience. We will now take the first question.
Hi. So you were saying that there are several different types of neurons in the gut already, that you're doing research to find, that you found out that several of these different types come from endoderms, or the mesoderm. So do you think that this implies that there's much more complexity to the gut, and especially the mesoderm gut, that increases in age?
That's a great question. So the question is whether with age, how does the complexity changes? We can gander a guess right now based upon the data that we have. We cannot absolutely give you an answer because those experiments are still undergoing. We think that these are adaptive changes. Our food patterns, our microbial patterns, our behavior, it changes with age. It's the same way that it changes during maturation. And we think that these are changes that are associated to adapt to the different functions of the gut. The kind of food that we eat, the kind of food that we can gustate, remember the gustatory function is mouth, are change over age because we cannot chew the same kind of food. We cannot masticate the same amount of food. The consistency of food bolus changes over age. So these are adaptive changes. That's what we think right now. And there are these adaptive changes that when they get fixed and become maladaptive, that's what is an extreme case of aging. But our current thought is that just as there is a normal expected behavior for every age, there's a normal expected behavior of the gut for every age, which requires a normal expected enteric nervous system for that age, an appropriate nervous system for that age. That's our guess right now, that's our hypothesis right now. We don't know whether it's true.
Hi, thank you for the talk. I was wondering, is there any ongoing research about gene modifications or mutations to the gut proteins or the microbiome, or like any genetic type of research?
Constantly. Our lab is the one that is also undertaking these things. We are interested in looking at how stress causes dysmotility, for example. Stress is associated with changes to the GI function. We are using a lot of gene-based therapies or gene-based modifications to understand what exactly is the mechanistic pathway for that. People have been trying to use gene-based modification systems for curing patients with Hirschsprung's disease. Hirschsprung's disease is an unfortunate condition that happens in about 1 in 5,000 kids, where kids are born without a functioning nervous system in their very distal part of the colon. And since this is not amenable to medicines because we have to treat the condition before the child is born, this is where people are thinking about taking the gene therapies next or cell replacement therapies. In terms of microbiota, this is an active area under consideration for many labs, including ours. The difficulty or the challenge is in finding the exact mechanistic pathway through which microbes talk to the gut. We actually published on this a few years ago, where we showed that changes to microbiota, people when they take antibiotics, they have significant changes to their gut motility. We showed that when we change the microbiota, it reduces neurogenesis in the colon. And if you supplement it with a particular drug, then you can still keep the nervous system intact. So those are things that we are trying to do. That's where the community is trying to go. Instead of just finding associations, which is easier to do, but then they are not replicative and they don't translate that well into therapy, the holy grail is in finding mechanisms. And once you find the mechanisms, then you can create the right drugs. Thank you.
Hi. What are your thoughts on food permeating, I guess, the gut barrier into the bloodstream? I think it's commonly kind of called leaky gut. I'm not sure if it's really accepted that it's a real thing or can happen, but I think people think it could cause autoimmune impacts. Yeah. So
leaky gut is a loaded term. The reason is because it is very easy to envision it. You have a gut barrier that is becoming leaky, so everything is coming in. It is also scientifically imprecise, and that's the reason why I'm going to segue a little bit away from that, and let's talk about the thing that we do see. So there is a loss of barrier function. So if you call it that instead of the leaky gut, it becomes a little bit easier, because it's not that the gut becomes leaky as a result of a number of things, but there are certain barriers that are aberrant, or they're lost, or they're less regulated. That does not mean that everything can come in. That means that there are certain proteins that could not come in to the same extent that are now coming in a little bit more. The question then is, what exactly causes loss of barrier function? People have a loss of barrier function when they have celiac disease and they eat gluten. People have loss of barrier function when they have significant inflammation of the gut. But in normal individuals, the gut is always leaky. That's how we ingest food. That's how we digest food, absorb food. So there is some amount of leakiness that is inherent, without which we will not be able to absorb food. Now, to the question that I think you're asking about, the autoimmune angle to it, surely, like in celiac disease, that causes it, but it's not the disease pathogenesis incumbent that's directly causing that loss of barrier function. It's a result of insults. It can be a result of many different types of insults. In terms of how much would it be involved in constant pathogenesis, remember the gut is a self-healing organ to the large extent. Right? I mean, you get an infection, the infection resolves itself, you're back to normal shape in a week without really taking any medication, because even you have a stomach flu. So since the self-healing nature of the gut regulates itself, there has to be a constant insult or a change in the cell type that is present in the gut that causes the pathology to manifest and continue. Those are different mechanisms altogether than having single insults. In terms of autoimmune conditions, let's take into consideration patients who have autoimmune diseases like systemic sclerosis. We are yet to find a very clear, concise explanation or a straightforward explanation for why is it that patients who have these rheumatoid conditions, these rheumatic disorders, they have significant GI disorders. We don't really find significantly large populations where they have loss of barrier function. So the question of how autoimmunity relates to gut function and dysfunction, it's still a very open question in my mind, and it's still something where there is a heterogeneity of mechanisms that are at play, and we can't easily come to one particular dysregulated or dysfunction and have a panacea against that, unfortunately.
Thank you. That was a good answer.
Thanks.
Thank you for your lecture. It's really, really informative. I'm curious if you'd be willing to speculate on what the supplementation of the GDNF could be on humans, if it's producing more motility in mice, and could you possibly expand on the potential benefits or non-benefits if that supplementation worked in the same way in humans?
There's a two-part answer over here. First, the bad news. The bad news is that there haven't simply been enough genome-wide association studies done on patients with motility disorders to understand how badly the GDNF gene is mutated in these patients. That's the bad news. We need more data. The good news is that in patients who have IBS, irritable bowel syndrome, we actually find that GDNF levels are way up high as compared to how much they should be. So this is one of those things where it's arrested development kind of phenotype. We also notice that patients who have, on the other side, flip side, patients who have obstructed defecation, chronic constipation, those sorts of disorders, there is a significant reduction in GDNF levels as compared to what would be normal. So it's a Goldilocks thing. You go on either side of things. We do observe that in patients, in human patients. One thing that I didn't put in over here is that the same two lineages that we see in mice, of course we can't do lineage fate mapping in humans, but we can take the adult markers that we can use to tell whether this is a mesodermal neuron or a neurocrest neuron. We find presence of both those neurons in the human gut, in resected tissues from humans. Not only that, but patients who show chronic constipation, we did bulk RNA sequencing analysis on them. So we took all the RNA from that tissue and compared that to healthy controls. And when we compared it, we saw that in the patients who have chronic constipation, the proportions of neurocrest neurons are way down. The proportions of mesodermal neurons are way up, suggesting that what we are seeing in our mouse models, where with age we are seeing these differences, really pans out to human systems as well. And GDNF is associated with that. Thanks.
Hi, I have a more so technical question. Have you looked at gastrointestinal peptide secretion and its effects on the enteric nervous system?
So there are various gastrointestinal peptides. So there's one which is CGRP, which is calcitonin G-related peptide. There is vasoactive intestinal peptide, or VIP. People did those experiments back in the day for physiological readouts. There have been varying degrees of success, and most of those experiments used to be done in guinea pigs, not in mice. There have been studies upon studies that have been focused on looking at what would be the effect of these peptides on the contraction or relaxation of the gut musculature. And they haven't really been updated because people have already done those. But that's the area that you're talking about is now more in vogue in trying to understand how the neurons communicate with immune cells in the gut. Because the neuroimmune crosstalk in the gut is supposedly significantly driven by these neuroactive peptides and neurochemical peptides. And that's exactly where, so VIP signaling and CGRP signaling is where the neuroimmune crosstalk is going. We have been looking at neuroimmune signaling in the gut in our lab as well, but that's not from the angle of neuroactive peptides. It's more from the cytokine release.
Thank you.
We have time for 2 more questions.
Do you see in the future a time when you'll be studying the effects that diet may have on this phenomenon of aging?
We have. We're currently looking at that, and we're seeing that if you model a deleterious diet, you cause accelerated aging. But the good thing is that if you stop that diet, it comes back to normal. We don't know whether that happens to the microbiota or not. I know that this was going to be a follow-up question to that. We think that it might actually go through a microbial pathway, but we are not so sure about it right now because we haven't done those experiments. But yes, diet plays a significant role, and people have been modeling various types of diets, especially what is called the Western kind of diet, which is very high in calorific value and high in fats. And people have been consistently noticing that and observing that those even a month-long exposure to that diet causes significant constipation in mice. If you stop the diet, it comes back. So 1 month of burgers and 1 month of salad is what I would recommend.
Hi, thank you for your talk. In the ways that I've studied brain-gut connection, it's been very intuitive and based off of body experience, and through indigenous and traditional knowledge. I think that's the way that it's been studied too. So this is a new way of knowing the brain-gut connection for me. And I'm wondering if you have any personal experience with experiencing brain-gut connection within your own body, or if anybody in your life has shared that with you.
Yes, every time I become hungry, I become angry. Oh, no, exactly. Not only angry, sometimes I get frustrated as well. No, it's a very interesting thing, right? I mean, there are so many. Consciousness is something that is very integral to us as humans, right? There are some questions that we have constantly pondered over. Where do we come from? It's one of them. What's the meaning of life? That's the second. Where's my next meal coming from? Is the third. And how is that meal going to make me happy? So happiness, satiety, on one side of things are a very rewarding experience. Nausea, anger, frustration, pain are the flip side of the coin. There is an integral network that goes to the gut, because if something really agrees with you, if you have a great meal, you feel very happy. If you have something that disagrees with you, it's amazing that the reflex of going from getting that first bite in to developing nausea can be instantaneous, right? So that instantaneous nature of things really tells you this, how integral these pathway is. What we don't understand is how this information is conveyed from the gut to the brain, because that feeling of I can't, this is disagreeable, I can't keep it in my mouth, I can't digest it, it has to go out, and then you vomit. That circuit is extremely quick acting, which means that there is a component of nausea, there is a component of lack of satiety, there is a component of pain, and there is a component of anger that all feeds in. So there are many different types of neurons that are activated, and that's the gut to brain. And then there is a brain to gut, which is it immediately sends a signal of eject. Now that all happens in a flash of a moment. And it's amazing how conserved that network is, that pathway is, for it to happen that fast. So yes, I mean, I think everyone in this audience would have a brain gut story to tell, especially if you have ever had food outside of your comfort zone, or even then not. But yeah, that speaks to me of saying, how relatable this science is to people.
This concludes tonight's presentation. Please join me in thanking Dr. Kulkarni.
Thank you.